Today we're going to discuss glycolysis and gluconeogenesis. First, we're going to have an overview of glycolysis. Glycolysis is a 10-step metabolic pathway that can function under aerobic or anaerobic conditions. Although it does not require oxygen, glycolysis is the first step in cellular respiration. Glycolysis, what it does is it splits glucose into two pyruvate molecules. Glycolysis can be thought of as having two phases: the investment phase, because it uses two ATP molecules, and the payoff phase. This happens in both aerobic and anaerobic states, so we'll briefly discuss both. In an aerobic state, pyruvate will enter the citric acid cycle and undergo oxidative phosphorylation, leading to the net production of 32 ATP molecules. In an anaerobic state, pyruvate changes to lactate through anaerobic glycolysis. Anaerobic respiration will produce 2 ATP molecules. The equation for all of this: glucose plus 2 NAD plus 2 ADP plus 2 pi yields 2 NADH, 2H, 2 ATP, and 2 water. Glycolysis occurs in the cytosol of the cells. With aerobic conditions, pyruvate will enter the mitochondria for oxidative phosphorylation. With anaerobic conditions, pyruvate will remain in the cytoplasm, where the enzyme lactate dehydrogenase will convert it to lactate. Now an overview on gluconeogenesis. In the second step, what we're going to do, gluconeogenesis is basically the reverse of glycolysis. We're going to talk about the steps where they're different. Where going backwards, they're going to be different. So in the second step, this differs from glycolysis in the conversion of fructose 1,6-biphosphate to fructose 6P. Then fructose 6P is going to be converted into glucose 6P. The last step differs in that glycolysis is converting glucose 6P to glucose. Here it is done with the enzyme glucose 6-phosphatase, which is found in the endoplasmic reticulum. In glycolysis and gluconeogenesis, 7 of the 10 steps occur at very close to equilibrium. It is important for an organism to conserve energy. Where there's excess energy, gluconeogenesis is inhibited, but when energy is required, it is activated. So it can be inhibited if there's an excess of energy, and it can be activated if energy is required. Now, aldolase is an enzyme. And we're going to talk about how it's involved in glycolysis. We're going to talk about how it's involved in gluconeogenesis. We're going to talk about the different forms of aldolase. So, in step 4 of glycolysis, the high-energy phosphates destabilize the fructose 1,6-biphosphate. This is when the enzyme aldolase comes into action. What it does is it cleaves or cuts the fructose 1,6-biphosphate into 3 carbon isomers: dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. At this point, this is the end of the work for aldolase in glycolysis. This is where it stops. That's it. It does nothing else in glycolysis. Below you can see this is glycolysis. Here's your aldolase. It's only in this one spot, and this is when it's doing that particular cleaving. It's going to make the dihydroxyacetone phosphate, and here's your glyceraldehyde 3-phosphate. So it's creating those two from your F16 biphosphate. So aldolase, like I said, we're going to talk about it in gluconeogenesis too. So here you notice it's right here. It's right by the fructose 1,6 biphosphate again. But it's going to go in the opposite direction. In gluconeogenesis, aldolase is the enzyme that catalyzes the joining of the substrate glyceraldehyde 3-phosphate and the substrate dihydroxyacetone phosphate to make the fructose 1,6 biphosphate. So it's taking this one and this one, joining it to create this one. Like I said, it's the opposite function of glycolysis. So where glycolysis, it takes it apart. In this particular one, it's going to put it back together. So there are different forms of isoforms of aldolase. So we're going to talk about those now and where they're found and where they're expressed. Aldolase A, embryonic tissue, muscles, and fetal glycolysis. Let's talk about that a little bit more. Aldolase A is found in embryonic tissue and adult muscles. Aldolase A is a crucial enzyme in the process of fetal glycolysis. Without adequate levels, fetuses could develop mental retardation, short stature, hemolytic anemia, and abnormal facial appearances. There is no known treatment for aldolase A deficiency. So if that was to happen, there's no known treatment. We can't fix it. We're going to go to C. We'll get back to B, but we're going to go to C right now. Aldolase C is in the brain. Aldolase C are arranged in a striped pattern in the cerebral cortex. This is also known as zebrin 2. Now let's talk about aldolase B. A deficiency of this enzyme would be shown in an infant's first exposure to fructose, or it can be delayed and have its onset in adulthood. Hypoglycemia occurs in liver. It could occur, and liver dysfunction could also occur. It can be treated by removing fructose, sucrose, and sorbitol from the diet. That's not an easy task, to remove all three of those from a diet. So what we're going to look at now is we're going to look at aldolase B and some of its transcription factors and why it may affect everything the way it does. So HNF1, HNF3, C, EBP, and DBP are transcription factors. In fact, they are all liver-specific factors controlling liver transcription. With aldolase B at the transcription start site, there are tissue-specific expressions on the reporter genes. They're active in the hepatocytes, but not in the fibroblasts. So these are all found in the liver. Their expressions are all in the liver, and this is where these transcription factors make their impact.